All right, so we're officially begun our iSeek live stream. We're, as you know, the International Society for Evolution, Ecology and Cancer. You can learn more about us on the web. Iseek.com, no, sorry, evocancer.com is our website. And we have meetings every other year where we bring together the evolution, ecology, cancer community, and have a very sort of interdisciplinary meeting. But we want to also have a chance for everybody to build community and for us to talk about important and interesting topics when we can't all be in person with each other. So here we are for a really exciting live stream. We have Amy Boddy, who is going to be talking with us about looking at cancer susceptibility across species, and Rob Noble, who will be talking a little bit more about sort of what's going on inside the body in terms of cancer susceptibility and differences in susceptibility in different tissues. So our general approach today is going to be to try to integrate everybody's questions as we go. And I will be sort of pulling in questions as they're relevant. So please, as you have questions or comments, write them in the chat right away. And that'll allow us to have a sort of more dynamic conversation. So don't be shy about putting any questions and comments in there. And then for the last half, we'll have an opportunity for more discussion as well. And so during the first half, I'll be mostly moderating the questions and comments and pulling things in. But then if there are sort of bigger questions that you have at the end, there'll be a chance for you to pop in and ask them yourselves. But I'll be sort of a heavy-handed moderator to kind of keep things moving along, and also to try to connect the dots with questions and comments that are sort of relevant to the flow of the conversation in the moment. So without further ado, I'm going to introduce our first wonderful speaker, presenter, Amy Boddy. And Amy's going to talk to us for about 7 minutes or so about her work. And then we'll have some discussion. And then after that, Rob will do the same and we'll have some discussion. And then we'll have some general discussion among the entire community. So Amy, please go ahead and begin. I'm going to spotlight you for
Great.
everyone here right now.
Okay, and then everyone can see my screen. All right. Well, thank you, Athena. I'm super excited to chat today about this work that our team has been doing. Been collecting data for over a decade to try and understand cancer variability and risk across species. And I just like to add that this is an amazing team that I'm representing today. A lot of this work is done by many, many different people. We're part of the Arizona Cancer Evolution Center. I believe it was Mark told us right here that when we got the grant and we started coming together as a group, he said we felt like the Avengers team coming together because we had evolutionary biologists and we had cell biologists and we had oncologists and we had veterinary oncologists as well. And so everyone came with their own specialty, and it was really and still is a really interesting and dynamic group. And none of this work would be possible without our collaborations. So, and what we were really interested in is why some species are better at suppressing cancer than others. If any of you have had a pet dog or a cat or a goldfish, you know that other species get cancer other than humans. But it was up for maybe debate how often cancer was prevalent in other species. And it really, really dates back to, I really love this work because it started with some really good theory. So it starts back to Peto, Sir Peto. He proposed something that we started calling lovingly Peto's paradox, where he said if a human is bigger than a mouse, and an elephant is this much bigger than a human, wouldn't we expect, if all else is being equal, and it's just the number of cells that an elephant has more of than a human. And since cancer is a cellular disease and its probability of getting that cancer mutation is caused by cells dividing, elephants have more cells than humans, therefore they should get a lot more cancer. But we don't see that, all right? So this is the expected cancer rate based on just all else being equal. That elephant has the same cancer suppression mechanisms as a human, it should get a lot more cancer. But that's not what we observe, and that's the paradox. And that is that the observed cancer rate, well, while we didn't know much about elephant cancer at the time when this was proposed, it suggested that there's more cancer defenses in elephants and other big long-lived species. And then it was discovered in 2015 that indeed they have duplications in really important tumor suppressor genes, suggesting that yes, they have enhanced their cancer defense mechanisms. I do want to say, because this gets miscommunicated often, probably Newsweek didn't do a good job here, that elephants do get cancer. It's nuanced. They just get less than expected for their body size. Okay. Had to say that. Okay. And so jumping into the promise of comparative oncology, this paper basically launched the field. I'm giving a little bit of a history here because this is where our minds were at when we started collecting the data. Is so say you have something like malignancy risk here, really high malignancy to low malignancy. You're looking at a various life history trait, body mass, lifespan. And we predict, based on Peto's paradox, that maybe as species get bigger and longer lived, they had to evolve extra cancer suppression mechanisms to mediate all those cells in their body. And therefore, that's really exciting because these are novel mechanisms of cancer defense. We have some ideas that they're probably different in these different lineages. And so it opens up a whole toolbox to explore cancer prevention mechanisms across the tree of life. This is all the theory. Does the data actually support it? And so we just published this paper this year in Cancer Discovery, and we have been working with a bunch of zoos to really ask these questions. Can these life history parameters predict cancer risk across species? And really, do bigger, longer lived animals get more cancer? And so we tested these theories. We worked with over almost 100 zoos. We have what we call a necropsy data set. That is, we're looking at animals that have died and they go on and get a necropsy and they look and find if there's any tumors or any malignancy at all in these species, and then they report it. We report on almost 300 animals, looking at reptiles, birds, and mammals. And they had to have at least 20 species 20 individuals per species. Okay, so what did we find? The first thing we found is cancer is ubiquitous across all of these taxa. Of course, there are some species that got very little or no reports, but we found that wherever we looked, at least there was cancer in at least most of these lineages. This is neoplasia. We looked at just neoplasia, and we also looked at malignancy. I do want to highlight here, this is something that's been observed in a few other papers as well. Mammals seem to be more vulnerable than other taxa to developing neoplasia or malignancy. And we can come back to asking why that is. I also wanted to highlight that there's just extreme variability across the tree. I know this image is a little overwhelming at first, but we're looking at both neoplasia and malignancy. And what I wanted to highlight here, these bars represent the prevalence, is just the extreme variability where some species are getting very little cancer while other species are getting a lot of cancer. Some of the highest cancer reported species are unfortunately these little ferrets and opossums, but then we have these species that represent maybe very little to no cancer, such as the dolphin and the white-faced Saki monkey. So this variation is really exciting because then we can test some life history variables across the tree to see if they're driving some of this variation in cancer risk. And so we did just that. We asked about body size and other life history variables. And the one thing, there's going to be basically two results I want to show here today, and then we can talk more in length about everything else. And so this is looking at malignancy, and what we're really interested in various life history variables. So this was gestation length, and this one surprised us. We found a negative relationship. So gestation length, the longer a species had a gestation or incubation length, they got less cancer, less malignancy. This is exciting when you think about it from maybe a cell division or a somatic maintenance type of perspective. And then, of course, the other variable that I wanted to show is body mass. So Peto proposed that bigger, longer-lived species shouldn't get more cancer. What we found was, in our previous studies, we found no relationship between body size and malignancy. We do find a slight positive relationship once we start controlling for that gestation length. And so maybe bigger species after controlling for gestation length get slightly more cancer. And so I think I'll end there. I want to say that this paper, we were just looking at life history variables. There's a lot of other avenues that we need to look at in terms of cancer risk across species, including genomic factors and the environment as well. So.
Great. Thanks, Amy. James has a question already here. He's asking whether there are differences in cancer risk across animals during the natural period when reproduction was most likely. That could be another way of asking the question.
During the natural, do you mean during it's the onset of cancer during their reproductive years?
James, do you want to?
Is that how you're?
Why don't I just unmute?
Yeah. Do it.
Yeah, no, basically, I mean, just sort of, if you were to ask the question, are there different, because the sort of the evolutionary prediction would be that natural selection will act to minimize the risk of cancer or really anything bad that's due to intrinsic errors like heart disease and so forth during periods when reproduction was most likely. So for a mouse, that would be no more than 6 months. So mice, therefore, actually have incredibly good cancer defenses for their needs, right?
Right.
And so that would basically mean that you'd have to correct the data for any changes in that are more recent, whether it's because they're in zoos or whether, dogs in captivity or humans under modern conditions, or even you can imagine like wild deer where we've taken away the predators. So
Yeah.
in other words, if you just think historically, and I know that's not easy because we don't necessarily, you almost have to guess for some of these, but I'm just curious if you could correct your data with that sort of framing.
Yeah. So this data obviously is from medical records, and so it's spotty. So we don't have age of all of the animals. When we do have age, we're able to look for when the cancer develops. It develops later, but not, everyone thinks that animals that live in zoos, they have an extended lifespan, and so they're getting cancer because of this extended lifespan. We're not seeing that. It's developing at, within the range of their normal lifespan. But yes, it's not early on, as you mentioned. And actually, in this data set, we removed most infants and juveniles because we really just wanted to look at adult cancer. And that's mostly because infants and juveniles have high mortality otherwise. So we didn't think we'd get really reliable numbers there on the cancer rates.
Cool.
Yeah,
Great.
but I
Thank you.
do actually want to say that this idea of there's a possum, or there's an island of opossums, right? Where it seems like they don't have any predators. And it's been studied from a life history perspective, suggesting that they actually have extended their, or slowed down their life history. And so that would be a really good population to go look at and see if maybe they get less cancer too.
They live about 50%. Well, they age 50% slower.
Yes.
Yeah.
Yeah. So I would love to look at them. Yeah.
You check out figure 1 in my book.
Okay.
There's an artistic drawing of it.
All right,
All right.
Michael, we've been messaging. Do you have another question about the estimate of average lifespan? If so, go ahead and ask it.
Yes, I do. That's a very impressive paper and great talk. In reading your paper, I just was wondering how you estimate average lifespan. For example, what would be the average lifespan of a field mouse?
Right. Yeah. So we used one of these larger life history databases, NAGE and Pantheria, to estimate the average lifespan. I have to go back and look at.
But what is a mouse's average lifespan? Do you recall?
I don't recall from. Yeah.
I guess my question is it the average lifespan in a zoo?
No, it would be
in the field?
Yes.
That's a big difference.
It is a big difference. And I think that's really interesting to point out, because we see that with zoos. There's a really nice paper that showed that wild versus, captive lifespans can vary, right? Sometimes they get extended at zoos. Sometimes they don't, such as moose or elephants are actually not so extended in zoos. And so looking at that variability and how often they get extended and how long would be really, really interesting to look at. So yeah, the wild estimates in lifespan are harder to get. We just
what would it be for humans?
What would
I'm just curious because in the paper it's hard to figure out what you mean by average lifespan.
Right. Yeah. We're averaging it from the life history databases, but I think even with that, we could do better, to be honest. Like, we need to do better at getting at these survival curves, and it's going to be different for various species, especially the wild lifespan versus the captive, especially species that have high predation rates, like you mentioned with the mouse, and suddenly there's no predators. That's going to be a very big difference.
Yeah.
Great.
Okay. Great. Thanks, Amy.
Yeah, thanks, Michael. So I encourage discussion in the chat about this. There's some already going on. And right now I'm going to invite Rob to come into this discussion and give some comments on what Amy has shared, and for you guys to talk among yourselves a little bit about this. And I'll be here in the background.
Sure, thank you very much. Yeah, I also find this a fascinating and very important study. Having read about Peto's paradox for many years, and the fact that it was proposed, 50 odd years ago, and this is one of the best tests yet of whether it really holds. And the fact that in fact you don't find null correlation, you do start to find these interesting patterns. You really need that much data to uncover this. So we've started to talk a little about the scientific implications, but I thought another thing here is just the sheer amount of work it took to get hold of this data. Can you give us some idea of how you did that, how you coordinated among all these different zoos around the world, what that involved?
Yeah. It was a lot. When I first started out, I would literally call zoos and ask, and then we'd have to fill out paperwork, and it was a long process because most of the time the zoos were amazing, but understaffed, very interested in all these questions, but it was like, what species would you want to look at? And maybe we could pull those records. And I was like, I want everything. And it was like, oh, we don't have the staffing to do that. But when we teamed up with ESCRO, the Exotic Species Cancer Research Alliance, we were able to have better traction getting a hold of zoos and getting all the paperwork. But it was still a lot of admin to fill out the specific calls and requests of paperwork. But yeah.
So this is
also we worked hand in hand with Northwest Zoo Path as well, that could help guide us in what zoos to contact. Yeah.
Okay, but this is data they would collect anyway, right? Because they always
Yes.
do
Yes.
necropsy. It's just
Yeah,
getting hold
the
of that
records were
data.
already there.
Yeah.
Yes, absolutely. The records were already there. It's just, you know, the power to get them, right? And so working with Northwest Zoo Path was a huge breakthrough in being able to connect with all these zoos and able to get their approvals. But the big thing that was also really tricky is we wanted denominators. That is, we didn't want the cancer cases. And that's what we had seen if you had done a literature search or something like that, is the cancer cases. And many people don't know that individuals in a zoo, especially if it's an AZA accredited zoo, they'll get a necropsy when they die, no matter if they had cancer or not. And so that was a really great way to look at the necropsy records and kind of standardize it and say, all these species have been looked at death, which ones had cancer or not.
Yeah. And as you said, that there's a complication as well, that they didn't necessarily die of this cancer, right? Do you have any feeling for how much the cancer that occurs in zoos was actually affecting the fitness or the lifespan of these animals?
I think it depends. Many people don't know that some animals in zoos get chemotherapy, right? And so sometimes it really impacts them. There's actually a paper that was published in Nature a few years ago looking at cancer mortality. And so these species were dying because of the cancer. This data set, we were able to look at all of it, basically, whether they died of it or if it was found incidentally in one of the necropsy reports. Would it have ever been a problem for the species? We don't know, right? But I think that's a really interesting question to ask, because sometimes, you know, we think about cancer and then it leads to malignancy and metastasis, and it can lead to mortality. But the other thing we can think about is how often species go on to get benign tumors and they never transform into malignancy. And that might be another interesting area to look at.
Because indeed, that could be one way of coping with
Yes.
humans,
Yeah,
right? If the
exactly.
malignancy is going to grow, well, sorry, if the neoplasm is going to grow, if you're going to get a tumor, can you make sure that it's benign? Because certainly humans, there are many forms of benign tumor, and maybe that's one way our bodies have adapted to cope with this process, is just to contain it.
Yeah, and I think that kind of adds into your tissue selection stuff that you'll talk about in a bit, is some
Sure.
species, various tissues might be better at making sure a malignancy doesn't happen, even if there is a neoplasia.
Yeah, that's something I'm very interested in. Yeah. Yeah.
Great. Rob, do you have any other questions that you want to pose to Amy, or any other sort of points of discussion that you wanted to get to before you give us your slides? And
I'm wondering if we could take that. There's a question just popped up in the chat, so maybe I'll let someone else have the questions.
Oh, do you think
sorry.
amphibians had less neoplasia because their limb regenerative properties?
Ah,
I love this
That's a
question.
good
great.
question.
There's one paper, and I don't remember it, but maybe I can find it. But they looked at, and I can't even remember what species that had regenerative properties, but they did radiation experiments in them. And they just, instead of developing tumors, they developed extra legs. And so, yes, I think regenerative properties is really important. And that's all my speculation. That's all I can say about it.
That's very exciting. And, and those kinds of things, I think, also just speak to, like, in this field of, evolution, ecology, and cancer, when you do see bizarre, crazy things like that, there's actually a sort of theoretical basis to go to for why that might be the case, right? You can speculate about, well, perhaps there's been selection at the organism level for continued sort of regulation of cell growth and cell movement into adulthood because of that regeneration, and that may confer cancer protection because you've got those mechanisms still working for making sure that the way that cells are developing and moving and reproducing, that actually can be self-organized still, right, into adult age.
Yeah, and I think terminal differentiation is really a stick, right? Like where these species that can do regenerative properties may like have a lot of cells that aren't stuck on this lineage. You can kind of get reprogrammed. So yeah.
And then, of course, that raises the question of why don't more species do that, right? There must be huge trade-offs
Huge
to
trade-off.
doing that. Otherwise, we would all be able to regrow limbs if they got cut off. So,
Yep.
great.
I mean, we do with the liver, right? But
Yep.
somehow our liver is able to do it without getting a lot of cancer as a side effect.
Great. Rob, would you like to take the floor now and share some
Sure.
of your thoughts and ideas about this and some of your results as well?
Sure. I will just switch to some slides here. So can everyone see my slides okay? Yes? Is that good?
Yes.
Yes, excellent. Okay. So what I'd like to talk about, so the main topic here is comparative oncology, but I know that as soon as it comes up, as Mike has shown, people start asking questions about what does this mean for humans? And ultimately that's something we care about a lot, is what are the implications here for understanding cancer risk in humans and how we might be able to lower that. So I'm going to talk about a little bit of research that I did actually with Mike Koshberg, who was asking the questions earlier when I was a postdoc with him a few years ago, looking at this question of how much cancer risk do we think our human ancestors would have got before we acquired our modern lifestyles. So the way we came at this, I found mortality data on various human populations. So here the blue curve is a modern US population. And you can see that mortality is low up until about the age of retirement. And then wouldn't you know it, that's when it starts to get much higher. And it increases approximately exponentially in later ages. And if you look at other historical data and other populations, so in yellow here, we have a historical Swedish population that was subject to a lot more mortality because they have much harsher lifestyles, not as good medical care and so on. You can see that their mortality goes up sooner, but the shape is generally the same. And all these gray curves are just variations of the same mortality function, just varying one parameter in that. So you can see we can explain all these curves with the same kind of law. And in red is a modern hunter-gatherer population. And again, you can see that their lifespan is shorter. Their baseline mortality is a lot higher, and they succumb to this much higher mortality much earlier. So this is something we can build in to a measure to a model of how cancer affects us across life. So we know how humans their mortality risk from all factors over life. The other thing we can build in is data on cancer incidence. So the black solid line here is showing us the US risk of getting cancer for the first time over lifetime. The thing to note here is this is a log scale. So here we have 10 per 100,000. So you have a chance of 10 per 100,000 of getting cancer when you're young. And then it goes up to 100 per 100,000. And then it's going up to 1,000 per 100,000. So about 1% per year at later ages. So this is the modern US curve in black, solid black here. Now, what I was also able to find was some cancer data for populations that have unusually low cancer risk. And here again, we have some hunter-gatherers in yellow. And then we have a couple of other contemporary populations in Thailand and Oman that happen to have very low cancer incidence. But again, if you just rescale the US curve among adults, so we're going to ignore, for reasons that Amy already touched upon, we're going to ignore children and infants. But if we rescale the adult curve to about 40% of the US incidence, then you can get a fairly good fit to these very unusually low cancer incidence figures. So maybe this gives us an idea of how low cancer risk can be in humans if we avoid things like obesity and alcohol and smoking and all the other ills of modern civilization. So what I did then is I just built these two factors, mortality and cancer incidence, into a very simple mathematical model. and use this model to predict what then we would expect of lifetime cancer risk given life expectancy. So the different colors here you can pretty much ignore. They're just feeding in different cancer death rates into the model, and it doesn't make much difference. So you can pretty much ignore the colors. But what matters is the relative cancer risk per year. So the solid lines here are based on the US cancer incidence rate. The intermediate ones, these are based on a 30% reduction, which is actually one way to account for that is if someone's about 19 centimeters shorter than the US average, then their cancer risk is about 30% lower per year. So it's good to be short from a cancer point of view. And the dashed lines at the bottom, this is reducing by 60%, which is consistent with the exceptionally low cancer incidence rates that I just showed you. So if you plot the prediction of this model for the US, it says that if life expectancy in the US is around 75, then lifetime cancer risk in the US should be about 40%. And that's in the right ballpark. If you're shorter and you're in the US, then it's only going to be about 30%. But if you look at pre-modern humans and you allow that they had this shorter lifespan, if you assume their lifespan was similar to contemporary hunter-gatherers at only around 45 or so, then what you find is their lifetime cancer risk would only have been about 10% because they never reached these later ages at which the cancer risk really increases. And as, again, we've already touched upon, what really matters from the evolutionary point of view is what is your cancer risk during your reproductive years? Because if you've already had offspring and they've already reached maturity and they don't really need you anymore, then evolution doesn't really care too much what happens to you. So if you look at cancer risk by age 40, then in the US, it's less than 2%. And if you look at pre-modern humans, we estimate it's going to be more like 1% maximum. And if you allow that their cancer risk was substantially lower than in the US, then you're down to barely 0.5% cancer risk by the time they reach 40. So these are actually very low rates, and this is much more explainable than a rate of 40%. So really, perhaps the reason we get such high rates is largely just because we're living so long beyond our reproductive years. The other thing we can look at is cancer risk across tissues. So if we look at this data set here, this was collected by Tomasetti and Vogelstein in a very well-reported paper some years ago. And what they did is they looked at a lot of different human tissues and they plotted the total number of stem cell divisions in those tissues. So that's the product of the number of stem cells in that tissue, as they estimated it, multiplied by the number of cell divisions per stem cell in that tissue. So this gives us the total stem cell divisions over a lifetime. And against that, they plotted the lifetime risk of cancer of these different cancer types. And what they found is a correlation. But that correlation itself doesn't tell you really what's going on here. I mean, it's interesting that there is a correlation, but what more can we say? So what I did was I came up with a mathematical model that simply assumes that the cancer risk should be proportional to the lifetime number of, or it should be dependent on the lifetime number of stem cell divisions, such that if you take the logarithm of 1, then it should be linearly correlated with the logarithm of the other. And what I did was, instead of plotting one regression line through all the data, I split the data according to the kind of tissue that the cancer was affecting. And what I found is that if you do that, you get a much better fit to the data. You explain far more of the variation than if you just plot a simple regression line. And what you see is there's actually a nice tight correlation within most of these groups of different colors. So each of these different colored lines corresponds to a different tissue type. And what's more, they all have similar slopes. And the slope is very close to 1, which is exactly what we would predict from the theoretical point of view. And the nice thing about this model is that you can then read off the risk per stem cell division in each of these tissues. And by the way, the blobs here, the 3 blobs are extra data points that we found for some neuroendocrine cancers that weren't in the original paper. So we could use these points to test our model and find indeed they lie pretty much where you'd expect them to be. And from this simple model, we can calculate the inferred cancer risk per stem cell division in different tissues. And what you see is there's enormous variation here. So we see that, for example, osteosarcoma, which affects bone, the risk per stem cell division of getting cancer in bone is extremely high. And the reason this is tolerated is because the cells in our bones don't divide very much. And at the other end, if you look at particularly the intestine in black here, colorectal risk, then you see that the risk per stem cell division in those kind of tissues is extremely low. It's actually 10,000 times lower. So what this suggests is that different tissues have evolved different mechanisms for controlling cancer, and some of our tissues are actually a lot more effective at it than others. And the other thing you can take from this is that if you look at each of these tissues, generally they lie below about 10 to the power of -2, which means that the risk of cancer in each tissue is no more than a few percent over a lifetime. The only exception being skin cancer, but this is skin cancer in white Americans who are exposed to a lot of UV light, which again we can discuss whether that's really something that our ancestors would have been. So I think what's fascinating here is how this this kind of analysis of cancer risk within the human body relates to the kind of data that Amy has been reporting for cancer risk across species. I think that there are quite a few parallels.
Yeah, absolutely. Just a few clarification questions that came up during your talk. James was mentioning that perhaps the data is different from some fossil data, and Indra was asking whether some of the difference that you saw for cancer had to do with the sort of technology for looking for cancer. So maybe you could just talk a little bit about the methods around the mortality data and the cancer incidence data.
Yes, so the mortality data is debatable for sure, and so in the paper we do discuss the kind of data based on archaeological evidence as well, which suggests that in fact mortality might have been significantly different in our human ancestors than we see in contemporary hunter-gatherer populations. What we wanted to do in this analysis is establish an upper bound. So therefore, we looked at a case that gave us that would generate such an upper bound. So we chose the mortality data such that it would tend to prefer higher estimates of lifetime cancer risk. And what we discuss in the paper is various reasons why this is likely to be an overestimate, and that actual cancer risk could be substantially lower than the upper bound we established. So really, here we were trying to be very conservative and saying what is the upper bound here, and the same applies to detection. Yes, it's true that detection rates will have changed over time, and it's also true that if we're looking at the cancer here, these aren't necessarily cancers that would have caused significant mortality or morbidity. We're only, like Amy, we're only looking at incidence here, and it's hard to say how much effect this would really have.
Yeah. Just a reminder to everyone, please put your questions in the chat. I'm not going to be just calling on people who have their hands raised because we're trying to make sure that the conversation is flowing in a very natural way. So please do that. And I want to, at this point, invite Amy to ask Rob any questions and for you guys to have some discussion. So Amy, here we are.
Yeah, thanks, Rob. That was super, super interesting. And I really enjoy thinking about within the body, like what's the Petos paradox? What's the elephant tissue within the body? And do we expect that there would be extra cancer defense mechanisms for some of these super dividing tissues where you see a lot of stem cells, but maybe you're not seeing a lot of cancer?
I think small intestine is the most fascinating one because the cancer risk in small intestine is so low. It's such a rare cancer relatively. And I know we've discussed this previously, and one of the big problems is it's so rare, it's really hard to study. It's really hard to figure out what's going on there. and it's hard to identify something obvious that's different between the small intestine and the large intestine. But even the large intestine, the cancer risk, given that the stem cells are turning over all the time, the cancer risk, and that the intestine is very large, is remarkably low. So I think studying the reasons for why those tissues are can control cancer so effectively is perhaps going to help understand this pattern.
Yeah, at this point, I also want to invite everybody who is a part of this Zoom webinar. If you have thoughts or ideas about this question, please put them in the chat. Why do you think that there are these differences across tissues? What do you think are the mechanisms that underlie them? This is an opportunity for us to, share ideas, and we can speculate about this and maybe get some traction on it. So please definitely put any thoughts you have into the chat as Amy and Rob are discussing this.
Yeah, thanks. And in leading into that, I think about in terms of, we've discussed it in our group, a bunch of organisms that go from being very big to actually getting smaller. And maybe they're keeping those big cancer defense mechanisms along with them, right? And then they get the superpower of being really good at defending against cancer. And I wonder within human tissues, if from an evolutionary point of view, once we started cooking and got more efficient at digesting the foods because of cooking, that our intestines have gotten smaller, and maybe it is carrying a legacy forward where there's extra cancer suppression mechanisms there.
Absolutely. Yeah, I haven't really thought about this in terms of within the body, but I know that there is some fantastic research looking in the comparative oncology fields. Particularly, Yamur Ertan has led a very nice study of what happened with dinosaurs when they transformed into birds, and whether that gave them a kind of hangover of cancer protection from being big dinosaurs. Yeah, I think that could be a factor in explaining how some of these digestive tissues perhaps that have shrunk compared to what they might have been in our ancestors, how they've therefore retained those protective mechanisms as they've got smaller, and therefore they get less cancer than we would have had as an ancestor. I guess it should be possible to model that using the same kind of model I've introduced at the tissue level. Yeah, there's another very nice question I just spotted in the chat, which is from Yannick, who says, is it known in non-human species that larger individuals tend to have more cancer? Because I explained that in humans, I think for every 10 centimeters you're shorter, you gain about 15% reduction in cancer risk. Yeah, it's particularly been studied in dogs, of course, and I know that's something you know more than me about, Amy. But yeah, we see this variation. Peter's paradox doesn't hold within a species. Within a species, if an individual is larger, if it lives longer, it will get more cancer generally. But when we look between species, we don't see that pattern.
Yeah.
Is that your take on it as well, Amy?
Yeah, for dogs, it's still up for debate for other species because we haven't looked at it really, right? And you would expect some species that are really sexually dimorphic, that the larger one is going to get more cancer. For example, gorillas, where the male species is so much larger than the females. Are they more vulnerable to cancer? That's an open question. We don't know.
Yeah, so I think I mean, one way of seeing this is that there's another paradox, which is called Simpson's paradox, right? And this is exactly a way of expressing this, that sometimes if you look within a subpopulation, you find a very different correlation to when you look between. And I think this is really at the heart of what we're doing here, is these kind of multi-level hierarchical analyses. I mean, is there any way that your data set was large enough to do that, to start looking at influences of body size and longevity in the data that you have?
We can for some. Like we have, again, sometimes it's only 20 necropsies per species. That's small. We got to, bump up those numbers to really tease it apart. We're starting to look at sex biases, Again, we averaged the body size. We didn't look at female versus male specific body sizes. Some of those numbers are so small that we might not be able to, but it is something we're kind of moving towards, and I think we're going to be able to answer at least within certain species. Those
Yeah,
types of questions.
Zach is mentioning here in the chat that body size is an almost perfect predictor of cancer mortality in purebred dogs.
Yes.
So that does suggest that there is something sort of basic and fundamental going on here with body size and cancer susceptibility, and that it's, you know, when you have the opportunity for evolution at the organism level to be operating for a long enough time, that you start to sort of attenuate that otherwise, you know, strong relationship that's there.
Yeah. And then Lauren Howard is asking, there's a huge difference in size between sandcats and lions. Yes, like we haven't really done that analysis, but it's on our minds thinking about it. We do have some more detailed data on primates, non-human primates. And what we find in our preliminary data, so this data set is from Species 360, so we've included more zoos, and what we find is there's a really strong positive relationship between benign tumors and body size. So the bigger animals are getting more benign tumors, but there's this really nice negative relationship. This is within primates, not within a species, but just within, you know, an order, a really strong negative relationship between malignancy. And I think that's really interesting too. So maybe they are, they're still having this like cell division, maybe mutation problem that we would expect in Peto's paradox, but there's some extra mechanisms that make sure it doesn't go to malignancy.
Yeah. Well, seems like it would be easier to evolve mechanisms to make growths benign than to completely suppress them, right? So maybe that's even a sort of intermediate stage while these cancer suppression mechanisms are evolving, is that you just are able to sort of switch these growths from a malignant path to a more benign path. And so they might be a little bit costly metabolically, or, you know, maybe they would affect organisms, you know, fitness a little bit still negatively, but certainly not as bad as if it were malignant. So at this point, I'd like to invite James to pop in because he's made a lot of comments, and he also has a bunch of ideas that are really relevant to this discussion. So James, let me add you in here.
Great, thanks. Yeah, I'm sorry. I like to kibitz on the topic. I can't help myself, but yeah, just the first thing I'll mention, sorry, is that you know, this idea that I think there's something going on that has nothing to do with the differential mutation accumulation, because so many pieces of evidence show that mutation accumulation is not linked to cell divisions. Yes, you can get some, but the vast majority of mutations in a lifespan, a neuron accumulates more than our hematopoietic stem cells, and somewhere in the same range as liver and colon and so forth. So it's not that, but I do think you don't get evolution without reproduction, right? And so stem cell divisions is probably providing opportunities for reproduction. But the other thing I think we need to consider with bigger bodies, particularly within a species, is that that's often regulated by the IGF pathway. So is it that there's more cells, or is it that there's just inherently different signaling? Like men can have more IGF signaling than women, for example. Now, those are just some quick thoughts. But this is a great subject, by the way. I loved your paper, Amy. Amy and everyone else on who's part of that paper.
Everyone else too. Yeah, and it's interesting because there's a question in the chat that kind of links to that. James, where it says, is the difference in body size between men and women sufficient to explain the longer life expectancy of women?
Yeah, no, it's from Marco De Dominici, who's actually in my lab. So,
Okay.
but Mark, I think it's an important point. But you know, is it? You know, I don't think, I mean, we'd have to ask someone who knows this better, but I don't think cancer, I mean, in other words, men die sooner of heart disease and probably just about everything else. So I think men just age earlier. So every age dependent disease is going to be a little bit more premature in men, damn it. But, you know, I don't, in other words, I don't think that cancer alone explains it, but I'm sure it's a contributing factor.
It's complicated as well because of other, I mean, hormonal effects on many cancer types and so on that differ between men and women. But I think that study based on height, I think they controlled for sex. So they looked at very large populations, and that's population average. But I don't think tall people should worry too much, because there's a lot of variation, of
Yes.
course, within both tall people and shorter people. So this is only an average from very large
Oh,
populations.
come on, Rob. Let the short people have something good.
Well, with all three of you here, I think it would be very interesting for us to just kind of touch on this big picture question also of, what is going on inside the body in terms of the evolution of these potentially very tissue specific mechanisms. Now, you guys know I like to think of the body as a sort of social system, right? That we're 30 billion cells that are cooperating and coordinating all the time to make us function, including having these mechanisms for detecting and suppressing cellular cheating. Now, if we look at the body as this sort of society, then you have division of labor, right? Different cells are doing different jobs, and they're reproducing at different rates, right? That's the sort of stem cell divisions. And they're doing jobs that have sort of different risks associated with them, right? So I'm wondering if for any of the three of you, if looking at the body as this sort of cellular society that has to regulate all of these sort of different jobs and different behaviors that are happening in different tissues, is a helpful sort of metaphor at all for framing some of these hypotheses or predictions, or even contextualizing some of the results that we already have around this.
Shall I give it? That's yeah. Essentially, the reason I did this a few years ago. What this then sparked for me was exactly this interest. How do we explain that? How do we describe this in terms of these multi-level models that explain what's going on within these tissues? Because within each of these tissues, we have somatic evolutionary processes going on. They have different microenvironments. I got particularly interested in spatial structure. So a lot of these tissues have different organizations. And I mean, that Talmsedi and Vogelstein paper, it's all cast in terms of number of stem cells and stem cell divisions, but it's actually really hard to identify what a stem cell is in most kinds of tissues, and that's certainly up for debate. So the way that tissues regenerate themselves and regulate themselves is very different. Tissues have solved it in very different ways. And I think by studying that both in the lab and in clinical studies, but also with these theoretical mathematical models, we can get more of a grasp of how evolution has solved these kind of problems. And one of them, I mean, if we look at the intestine, spatial structure is a very obvious feature there. That the stem cells in the intestine turn over a lot, but they are organized into these small communities, and that makes it much harder for advantageous mutations to take over and then to spread beyond those little crypts.
Yeah.
I think we almost have to start with the evolutionary perspective, this is what everyone's been talking about during this time, but is that let's say that basically we evolved to not die prematurely, right? We evolved to reproduce, and anything that impeded that would be selected against. So you can imagine if the intestine created more of a problem, well, evolution just had to work that much harder to create those mechanisms so that the intestine, we wouldn't get cancer that limited our reproductive success in the intestine any more than we would in a tissue that might have far fewer cells, or for whatever other reason might be easier to not get the cancer in. And the same thing applies to heart disease. I mean, if you could look at the curves for everything bad that happens to us in old age, and they're all pretty on top of each other. And there's only one explanation that can really explain that, and it's the evolutionary one.
Yeah.
I think what's interesting though is that there are predictions here. So as Amy said, that has predictions about the evolutionary history, say, of the colon, that could be testable by looking at, okay, what was our colon like 100,000 years ago, 1 million years ago, 10 million years ago, and to see if we can find whether that evolution is consistent with that kind of explanation. Sorry, Amy, you were about to come in as well.
No, it's really interesting. I was thinking when it comes to like neighborhood models of it, I go back to what James has proposed in his book, where like your neighbors matter a little bit, right? So if you're in a tissue that is dividing all the time, and probably a lot of your neighbors have mutations too, right? You're accumulating stuff from dividing all the time. It might be harder to get a competitive edge there. You're in like an elite area where like having that good mutation that like, you know, gets you a higher fitness is much harder, right? Than these other dividing tissues. So it's just like ramping up, but your neighbors actually matter a lot, right? In terms of policing you and competing. So yeah, it's highly social, but it's also this competitive environment. And yeah.
Great.
Totally agree with Amy on that. In fact, it's actually, you know, if you could imagine, that could explain this accelerated cancer incidence in old age. Because once your neighbors start getting crappy, they're no longer able to keep the bad ones in check.
Yes.
And that's feed forward, because, you know, the more crappy neighbors, the more expansion of the more, even if not malignant, just sort of, you know, less social cells, then you're going to get even crappier neighborhoods and so forth.
Yeah.
And once the neighborhood goes downhill, the rules no longer apply. Basically, you're no longer living under covenant control.
There goes the neighborhood.
Yeah. Oh, by the way, it's 5 to 7 trillion cells in a human. I think you had said 30 billion. I don't know if you were talking about a particular tissue or something, but
yeah. Thank you for that clarification. James, I'm gonna thank you for joining us for the end here. And Rob, Amy, any final thoughts that you want to share with everyone?
I'm really excited about this work. I think this paper was really fun to put together. I think with larger numbers, we might see some different patterns emerging. So, yeah, there's caveats with all of this work when it's, you know, limited data.
This is what I was going to ask. Yeah, next. I mean, what would your dream data set be? You know, if you could have anyone send you some Excel spreadsheet, what is the data you'd most like to get hold of to answer these questions?
I think it exists too. So yeah, I think the dream data does exist out there. It's a coordination problem. And so getting medical records, animals get really, really amazing medical records. I think one other thing I want to point out is that the zoos and the veterinary community do an amazing job of protecting their patients. Just like humans need protection, animals do too. And so there needs to be checks and balances in place on how we access this data. So it does exist. The perfect, maybe almost perfect data set is out there. It's just getting large enough numbers really in order to retest some of this stuff. Yeah.
Great. Well, Amy, Rob, thank you both so much for joining us. This was wonderful. And thank you to everyone who joined us as well on this link, especially those of you who helped to make this such a dynamic conversation by adding your comments and your questions. We will be posting this on the iSeq YouTube channel, and we'll share it on social media as well. So this will be available for everybody to see in perpetuity. And we'll be keep your ears and eyes tuned, because we'll be doing this again. And we'll be sharing additional resources as well. And we're looking forward to seeing you all in person when we have the chance to do that again. But until then, we'll be here doing live streams. And thank you all again for joining us. Amy, Rob, it was great having you, and lovely to see all of you. Until next time.
Thanks. Bye, everyone.